In this video, we're going to talk a little bit more about chiral molecules, and we'll look more at absolute and relative configurations and how these are related to physical properties, in particular optical activity. Now, back in the early 1900s, scientists focused on a molecule called glyceraldehyde, and notice we have a chirality center. Now, this is important not just chemically, but also biologically. On paper, you can draw R-glyceraldehyde and S-glyceraldehyde, right? We can assign RS configuration, and we can see that these are two different compounds. In lab, if you have two different samples, you can see that one of them rotates plane polarized light in the dextrorotatory direction, and the other one is levorotatory. So we have minus and we have plus. But it's not obvious whether S goes along with plus and R goes along with minus, or does R go with plus and S goes with minus? So this can't be determined easily. So this was back in the early 1900s, and all of the tools that we have today were not available back then. So the scientists did something that was really, really scientific, and they guessed. They said, let's assume that our glyceraldehyde is plus glyceraldehyde. So they assumed our glyceraldehyde is plus glyceraldehyde. Therefore, S would be minus. Great, who cares? This is only one compound. But from this assumption, scientists went on to assign configurations of thousands of other compounds. So how did they compare compounds? We just talked about some reactions that occur not at a chirality center, and if the reaction doesn't affect the chirality center, then if we know what happens in the reaction, we know what to expect in the product. And so this one would still have the same relative configuration, right? It has the same relative configuration because the relative configuration means comparable groups occupy the same relative position in space. The same relative configuration means comparable groups occupy the same relative position in space. So by carrying out known reactions, they would know what the relative configuration is. Then by measuring a sample of this in a polarimeter, then they could measure whether it is plus or minus. Well, this was done for many, many, many compounds. And for example, R plus glyceraldehyde was converted to R minus lactic acid. S minus glyceraldehyde can be converted to 2R3R plus tartaric acid. So notice we have the same relative configuration here, and because of priorities, these both happen to be R. We have the same relative configuration here, and because of different priorities, here it's S, but over here it's R. That's not the important part, really. So it was known that they started with a compound that rotated in the plus direction, and it was known that they had glyceraldehyde. It was assumed that it was the R configuration. It was measured that the lactic acid rotated in the minus direction, and then based on the relative configuration, they would assume, based on this assumption, that this would still be R. So by carrying out this process, thousands of compounds were determined as RS relating to the plus minus, all based on an assumption. Now, in 1951, X-ray diffraction of tartaric acid that rotated in the plus direction was proven to have the 2R3R configuration. So scientists got lucky, right? It was the correct assumption, and every other molecule that was assigned its configuration relative to glyceraldehyde then was correct. And this is a really important thing because this was 50 years of research, 50 years of journal articles, 50 years of making medications, 50 years of writing textbooks, 50 years of labeling bottles of reagents. And so if in 1951 they had figured out that, oops, I made a mistake, that would be so many things that would have to be changed. All right, so I just wanted to share that with you, and I want you to be able to keep in mind absolute configuration refers to R or S based on the Konigold-Prelog rules. Relative configuration is a comparison of the orientation of similar groups in space. So relative configuration is about looking at two molecules with similar groups in the same space and deciding whether they are the same or opposite. Absolute configuration means you look at a structure written on paper and you assign priorities and assign R or S configuration. And then we also have the optical activity, which can rotate in the plus or the minus direction. Remember that the plus minus is not directly related to R or S. So it's not directly related to the absolute configuration or to the relative configuration.